Today's topic is plant cell and tissue culture. I am going to tell you in simplest way what is plant cell and tissue culture. To understand that, we have to understand what is plant cell first, then only we can understand what is cell and tissue culture, right? It's a very basic thing of plant biotechnology. Plant biotechnology starts from the plant cell and tissue culture only, right? And I hope you have basic understanding of plant cell at least, right? So, as we know that plant cell and animal cell, the basic difference is there, one basic difference, right? And that is, a plant cell used to have this is general structure of one plant cell and this is the structure of one animal cell. Yes, what is the difference here? The basic difference is here two coverings are there. This is called as cell wall and this is called as cell membrane, right? So, in plant cell two covers are present, two boundaries are there, cell membrane and cell wall, and in case of animal cell we have only one cell membrane. Yes or not? This is the very much basic difference in between plant cell and animal cell. If any kind of cell that is having cell wall, we will consider it as plant cell only, right? It may be fungi is also having cell wall. Yes or not? Right? So, the basic difference in between plant cell and animal cell. What is cell? Now, the structural and functional unit of life. We know that basic difference, right? From our 9th to 10th standard itself it is mentioned that, right? The basic or the fundamental unit of the functional and what is the definition? One definition is there no of cell. The structural and functional unit of life is called as, or body is called as plant or animal cell. So, this is the structure of plant cell, this is the structure of animal cell. We are going to discuss about the plant cell only, not animal cell here today, right? So, when the plant cell start cell division. Cell division you understand?
Yes, sir.
Yes or not? Multiplication of cells. One cell become two cells. Yes or not? One cell become two cells. That is called as cell division, and generally that is mitosis. That is mitosis. So, one cell that is 2n can produce 2n plus 2n, no 2n plus 2n, 2 cells. Similar, just like mother cell, daughter cells will be there. No difference in this cell and that cell. Yes. Then further cell division will be there in same way, 2 become 4, 4 become 8 and formation of many cells like this. Yes. Formation of many kind of cells. Right. All these cells originated from this single cell. Yes or not? This single cell undergo many cell divisions and formation of a group of cells. Formation of a group of cells, right? Then this group of cell is called as tissue. It's called as tissue. So, single cell, with single cell many cell divisions we got group of cells. Group of cells of, as we know that how it happened, group of cells of same kind of originated from the same cell. Yes or not? Originated from the same cell. So the group of cells of same origin, right? So since origin is same, so same type of structure will be also there, right? So group of cells of same origin, same structure. Since these are of same origin, same structure, they will perform same kind of functions also, right? So a group of cells of same origin, same structure and same functions. These group of cells are called as now tissue. Got the point? You understand the difference between cell and tissue? What is the topic today? Today's topic is plant, cell and tissue. Forget about culture right now, right? So plant cell, a cell that is having one extra boundary other than cell membrane, that we used to call it as cell wall, right? So a cell with a cell wall is a plant cell. This is the basic difference in between animal cell and plant cell, right? Animal cell without cell wall. So when this cell is undergoing many kind of many cell divisions, that is basically mitosis, produce number of cells. These group of cells of same origin, same structure and same function. Now these group of cells are called as tissue. Is it okay? So plant cell, this is plant cell. Plant tissue, this is plant tissue, right? I am not drawing here double layers here, right? Actually cell wall formation also starts after cell division immediately, right? So cell wall formation also starts. So now these group of cells are called as tissue. It is clear what is plant cell and what is plant tissue. Now what is culture here, right? Culture means, a meaning is there of culture. Culture means growth and development. Basically culture means growth and development. It's not like it's a South Indian culture or North Indian culture, or right? Culture means here, in this case, culture means 2 points are there: growth and development, right? Of what? Culture means growth and development. Growth and development of what? Growth and development of plant cell, growth and development of plant tissue, right? So what is today's topic? Plant cell and tissue culture, right? So the meaning of a culture is growth and development, but the definition is, what is definition of culture then, right? That is quite different. What is that? This is the point number 1 to define culture, right? Point number 1 is this only: growth and development of what? Of cell and tissues. Point number 2 is on artificially prepared. On artificially prepared what? Nutrient solution, right? On artificially prepared nutrient solution, right? You might have seen that nutrient solution here. What we call nutrient solution in general, we call this nutrient solution is medium. You are making medium, yes or not? That nutrient solution here we used to call it as nutrient medium. Nutrient medium. Medium is plural, singular. Plural is called as media, not mass media, multimedia, social media, not that, right? Be clear here. So here, so we are discussing about what is culture here. Culture means growth. Point number 1, growth and development of plant cell and tissues. Point number 1. Point number 2, where? On artificially prepared nutrient solution, that is called as medium here. Yes, right. Point number 3, under the controlled conditions. Under the controlled conditions. What is controlled conditions then? Yes? Under the controlled condition. And that is also, controlled conditions are also called as laboratory conditions. That is also called as laboratory conditions. And that is also called as, sometimes we used to call these as under in vitro conditions. That is also called as in vitro conditions. Right? So, what is the third point? What we are discussing now? Culture. So, point number 1 of culture is, to define culture is growth and development of what? Plant cell and tissues. Right? On where? On, sometimes under also. On or under artificially prepared nutrient solution. Right? Inside. That is called as medium. Where? Under the controlled conditions, or under the laboratory conditions, or under the in vitro conditions. In vitro conditions. Right? These things are, what are these controlled conditions? Right? Or what are these laboratory conditions, or what are these in vitro conditions? Right? To maintain temperature. Very good. Very good. Very good. So, under controlled conditions, condition number 1 is, C is perfectly under controlled temperature, because plant cell can grow, right, at certain temperature conditions only. Right? So, what is the optimum conditions? Very good conditions where maximum growth can be possible. That is called as optimum temperature. Condition number 1, optimum temperature. What can be other 2 or 3? Controlled conditions. So, proper temp, in other words we can say proper temperature. Number 2, since this is plant cell we are growing, so for proper growth and development of plant cell, what we need? We need proper light is needed. If we are culturing animal cell, then light is not needed there, because for plant cell used to do photosynthesis, chlorophyll is there, right? So, for proper growth we need proper light. And the third condition is called as proper humidity, right? The moisture in the air or surrounding, right? So, if we can maintain these proper conditions, it means if we can maintain a place where we can maintain proper temperature, proper light and proper humidity, that is called as optimum, where the plant can grow in minimum time period, maximum growth can be possible. That is called as controlled conditions. Where these controlled conditions we can make, that is possible inside the lab only. Yes or not? Not under natural conditions. That is why it is also called as under laboratory conditions. And if we are maintaining these conditions inside the laboratory, so that is also called as in vitro conditions, right? In vitro conditions. So, let us define again what is culture here. Culture means, point number 1, what? Growth and development of what? Of cell, plant cell and tissues. Point number 2, artificially prepared nutrient media. Clear? Right? Where? Point number 3, under the controlled conditions or in vitro conditions. And controlled conditions means optimum temperature, optimum light and optimum humidity. If we can provide these facility, then the plant cell and tissues will grow very fast. Fast in the sense, better growth will be there, proper growth will be there, right? Minimum time period, maximum growth can be possible. That is called as culture. You understand the meaning of that? Each and every term here, right? What is plant cell? What is tissue? What is culture here? Plant cell, a cell with cell wall is plant cell, right? This is the basic thing, right? Tissue, group of cells of same origin, same structure, same function, right? This group of cells, this group of cells, this group is called as tissue, right? Culture means, right, the growth and development of, culture means come backward again, right? The growth and development of plant cell and tissues, right, on artificially prepared nutrient solution, that is medium, under the controlled conditions. That is called as plant cell and tissue culture. Got the point? Right? Sometimes they can ask what is cell culture. Then what can be the definition? What is cell culture? Only cell culture. Simple, no? The growth and development of plant cell on artificially prepared nutrient solution. Yes or not? Under the controlled condition. That is called as plant cell culture. Simple. What is plant tissue culture? Growth and development of plant tissue, artificially prepared. That is called as plant tissue culture. Got the point? 2-3 more points also can be here, right? For example, you might have in your you are in second year or third year? Second year. Okay. Plant cell and tissue culture part is over. System physiology. Okay, fine. Right. Sometimes some more words you will get. What is organ culture? What is organ culture? Sometimes ovary culture. Right. These words you will encounter in next semester. Right. Organ culture, ovary culture, even pollen culture will be also there, right? Pollen culture will be also there, right? Even embryo can be cultured, right? You will have many words like mariculture, maricloning, micropropagation, right? So, what is the difference here? See here, pollen culture, organ culture, ovary culture, embryo culture, right? So, basically these are organs, yes? Organs of the plant. Plant used to have few organs only, yes? What are these? Root, stem, leaf, flower, seed, yes or not? Fruit, yes or not? And if we go deep inside, then we can get anthers, pollen, yes or not? Inside the embryo, we can get ovary and embryo sac, we can get even endosperm also, right? So, different these organs we can also culture, right? For example, organ culture, same definition. What is the difference here? Organ only. Yes or not? Organ only. Yeah. So, culture. What is organ culture? The growth and development of plant organ. Right. Point number 2. Under the controlled condition. That is called as organ culture. It may be ovary. Same way. Prepared solution. So, what is the difference here? What we are culturing? Yes or not? What we are culturing. And the what we are culturing here, it is pollen, here it is organ, here it is embryo, here it is ovary. This is called as explant. What is explant? Then what is explant? Yeah, exactly. The piece of plant, it may be stem piece, it may be root piece, it may be flower part, it may be seed also, it may be ovary, embryo, pollen, and anther, anything, right? The piece of the plant which we are taking or using to initiate the experiment of culture, that is called as explant. Explant. The part of the plant or the segment of any plant part, right? It may be piece of root, it may be piece of stem, it may be piece of leaf, it may be flower, it may be seed also, right? Anything which we are, that is live, that must be in live condition, not dead, right? It should have live cells, which we are using to establish the cultures, to initiate the experiment. That piece of the plant is called as explant. Got the point? Okay, some more points are there here related to the basics of plant cell and tissue culture. So now what we understood, what is plant cell, what is plant tissue, what is plant cell culture, what is plant tissue culture, what is organ culture, what is ovary culture, what is pollen culture, what is anther culture. If you are culturing anther, then it is anther culture. Got the point? What is explant? We also understood in this what is medium, right? Yes or not? That much we things we understood here. So this is very much basic thing of plant cell and tissue culture. One more word is there. We understood what is in vitro also, no? Yes? In vitro. In vitro means basically this word came from the word vitreous, right? In glass, something we are growing in the glass. Yes or not? So it means something we are growing in the laboratory conditions, that is called as in vitro. One more word we are using in plant cell and tissue culture, that is called as ex vitro, right? And we know that one more word is there, that is called as in vivo. Yes, in vivo means, okay, in vivo means under the natural conditions. This plant is growing in the nature without our control, that is called as in vivo. In vivo means under the natural conditions without any type of our control, right? In vitro means fully in our control. We told no, under the controlled conditions. What are these controlled conditions? Proper temperature, proper light, proper humidity. If these three things we can control, then it is called as under the controlled conditions, under the laboratory conditions, and that is also called as in vitro conditions, because there we are controlling the temperature, humidity and light, that is in vitro conditions. What is ex vitro? Ex vitro means at certain extent we can control these three conditions, at certain extent only. So something natural control, something we can control, right? That is called as in between these two, under not fully under in vivo conditions, not fully under in vitro conditions, that is called as ex vitro conditions. And here our example is we are maintaining these conditions in the greenhouse conditions, right, where we are hardening our plants, right? We are making micropropagated plants, we are making here, we are making tiny plants, right? These if we are directly transferring to the field, they will die. So first we are hardening them, adapting them step by step, putting somewhat harsh conditions, then more harsh conditions, then finally more harsh conditions, and finally these plants will go to the farmer's field. So these little bit controlled conditions we are maintaining into the greenhouse, and the greenhouse conditions we are calling it here as ex vitro conditions, not fully under natural, not fully under the laboratory conditions, that is called as ex vitro conditions, right? So this is a very basic thing in simplest way about what is plant cell and tissue culture. Any question, any doubt, most welcome, right? Anything which you feel that this can help, any doubt? Finish it.